Okay, so in terms of our actual sort of immunology stuff here, so an antigen, very important, again, definition to know. So it is a molecule that is capable of inducing an immune response. So we've got our self and our non-self antigens. So on our pathogens, they will have their own little antigens. If we're thinking of a bacteria, um, maybe just a little protein on the capsule, or if we're thinking about the virus, like a spike protein, that is the antigen. It's just like a little, I like to think of antigens as name tags. It's just like a little name tag basically identifying what that pathogen is. And if you think of a self antigen, so like your MHC markers, um, these are again little name tags that say, hey, like I belong to the body, basically. So your MHC markers are your MHC1 in particular are basically your self antigens. Um, so they are found on all nucleated cells. So be aware that they aren't on, certain cells like red blood cells and things like that, but they have their own little, like they've got their ABO system. So there's still ways of identifying cells there. Um, but your MHC1 are essentially, yeah, your little name tags. And the way that they work is, and I think this is important to know. I remember I didn't know um, this sort of second dot point here. I didn't know it until we were kind of further into the area of study. And I think to me, it helps everything click a little bit more, especially when you think about the difference between cytotoxic T cells and natural killer cells. Knowing what MHC1 actually does, as opposed to just being a self antigen, for me was really helpful. So just bear that in mind as we talk through them. Um, so with the MHC1, I like to think of MHC1 as basically little windows. I think I might elaborate on this when we get to natural killer cells and stuff like that. But they basically show immune cells. So I like to think of yeah, so cells as houses, your MHC1 as windows, and your immune cells as police. So your immune cells go around and they just basically double check everyone's antigens and make sure that there's no foreign antigens hanging around that they need to be a little bit wary of. So the way that they do that is through these MHC1 markers. So that's why I like to think of them as little windows, because basically the police, so the immune cells, they run around and they look in everybody's windows. So they look at these MHC1, and MHC1 shows what proteins are being made in the cell. So they present peptides derived from cytosolic proteins. So your cytosol, proteins that are hanging around in your cytosol, they take like little bits of that and they put them on their MHC1, and then that shows the immune cells, this is what I'm up to inside of my cell. So that's why I think of them as a little window. So then if something is going a bit dodgy in there, in your MHC1, then the police can realize and then they can sort it out. Yes, that's what I like to think of it as. So obviously, if you're making the proteins that you're meant to be making, that's how MHC1 works, is basically a self antigen, a self marker. It tells the immune cells, I am a cell from this body. I'm meant to be here. I'm not doing anything wrong. Your class 2 are a little bit different. They are associated with antigen presenting cells. So not every single cell has MHC2, only specific immune cells have it. And these will be used, like I was going to say window again, but not really window. These will be used basically just to show when you've got a foreign pathogen hanging around the body. Okay, so we'll show a little picture. So here is this idea of self antigens and non-self antigens. So your T cell, which we'll get into in a little second, is a really important immune cell. So they go around and they, can check what's going on. And it's this idea that they have different receptors. If there's a theme for immunity, it's receptors, this idea of things being complementary, matching up, detection, all that sort of thing. A very, very important sort of theme of immunity. So your MHC1 markers, as we mentioned, are your self antigens. And then your, stuff that might be on a pathogen is your non-self antigen. So T cells are able to basically detect what belongs in the body and what doesn't. Yeah, so hopefully that makes sense. Okay, so to get us sort of thinking, so when a virus attacks or infects a human cell, what occurs within the cell? So again, think about your viral particles. Think about what our definition was. So an obligate intracellular pathogen. I was thinking parasite. Yeah, obligate intracellular pathogen. So we're going in a cell and we're basically hijacking that machinery. So we are undergoing transcription, we're undergoing translation, and that cells, our human cell, will begin to become basically a virus making factory. So MHC1 markers, what they present on the outside of cells. So hopefully you guys are all thinking of the little analogy of a window. Okay, I don't know if it's helpful. It was helpful for me. If you think it's stupid, don't use it. Think of something else. But yeah, I just like to think of them as a little window. So they present your little proteins that your cell is making. Again, just a way of showing the immune cells that you're sort of doing the right thing. If you think about it, with a virus, if you're infected with a virus, what you would be making, if you're making viral proteins, you're going to be essentially displaying that on your MHC1. So that's this idea of how the immune system detects cells infected by viruses. Your MHC1, so your windows, they'll be displaying your viral particles. So you've got, I don't know, a home invasion, right? The virus is a home invader. I guess they come in, they start invading, and your windows are still open, and the police come around, your immune cells come around, and they can see through the window that there is someone invading your home, and then they sort it out from there. So that's the idea of how immune cells detect when cells are infected by viruses, via their MHC1 markers.